The aim of this presentation is to show how Western blotting and immunocytochemistry can be used to demonstrate the SorLA and CLC-CLF1-mediated downregulation and lysosomal degradation of the CNTF receptor in human cell transfectants.
This method can help answer key questions regarding the turnover of the GPI-anchored CNTF receptor upon its co-expression with the endocytic receptor SorLA and exposure to CLC-CLF, which interconnects the two. The main advantage of this technique is that it does not involve radioactivity and is suited for membrane proteins which exhibit only a small degree of degradation and new synthesis in unstimulated cells.
Use stable clones of human embryonic kidney 293 transfectants expressing MYC-tagged CNTF receptor, or co-expressing MYC-tagged CNTF receptor and SorLA. Seed equal numbers of cells in 2 4-well plates. Culture the cells in full medium, which is DMEM supplemented with 10% FBS, penicillin, and streptomycin, in a 5% carbon dioxide incubator at 37 degrees Celsius. Once the cells are 50 to 80% confluent, remove the medium from the wells. After washing once with Jolbeco PBS, add fresh full medium with or without 10 nanomolar CLC-CLF1 to each cell type on both plates. Then, incubate the cells at 37 degrees Celsius. After 0 and 5 hours of incubation, recover the medium and store it at 4 degrees Celsius in 1.5 milliliter tubes. Then, add 1% Triton